Welcome back. We're going to take another look at the production issues in making biological molecules like insulin and penicillin. And we're still in the enabling science we call biology. If we're looking at penicillin and insulin, these are medicines, and some of the issues about how to engineer them and how do you make them in large quantities for safe use in humans, for example. Penicillin, as a reminder, it's an antibacterial. It's an antibiotic, as I mentioned. Insulin is a hormone. It's used to transport glucose from the bloodstream into the cells. If you don't have insulin, either if diabetics don't make insulin, or they don't make the right type of insulin for some reason, they're not able to transport glucose into the cells, and they could be starving. And so one solution in that case is to make insulin and inject them, and diabetics can therefore now transport glucose into the cells. And penicillin, and both of these are miracle drugs, really. It's just amazing as to imagine life without antibiotics or without insulin. So there are similarities in these two molecules. The similarities are both of them. Penicillin, for example, is a complex molecule containing carbon, hydrogen, oxygen, nitrogen in different proportions, right? Is it simple? It's not. You can call it simple compared to, say, insulin perhaps, but penicillin is still complex, say, compared to ethanol or methanol and so on and so forth. It's still complex. Insulin is, of course, a lot more complex. It is a polymer made of individual amino acids that are strung together. The question really is, and I can say, I mean, if you draw the individual structures of amino acids, you'll see the carbons, hydrogen, oxygen, and nitrogen, but you'll notice that this is, of course, much more complex than a structure like this, if you will. So the question really is, in both of these cases, for insulin, if you remember, we talked about using bacteria to make it. In penicillin, if you recall, we used mold to actually make the penicillin also. The question we can ask to the engineer or the biologist is it possible to make them using synthetic organic chemistry? Yeah, but it is extraordinarily complex, and biological synthetic machinery is so advanced that we are really unable to match it in the lab. So what we do as engineers is figure out how to use the biological machinery in order to make these complex molecules in the vessels, in the reactors that we sort of design. And so maybe in principle it's possible to certainly make penicillin, but even penicillin, for example, if there is a more the biological method can actually be much more efficient and safer to make it. Amino acids, as I said, you can imagine making different amino acids and then using synthetic organic chemistry to string them together, but using the biological processes turns out to be much more efficient. Yes, you can make individual covalent bonds, peptide bonds, and so forth, and using synthetic chemistry, but being able to use the biological processes to make it is definitely much more efficient. So yeah, you can synthesize insulin using synthetic organic chemistry. Not really. I mean, it is not something you would want to attempt, perhaps just as a curiosity of trying, maybe just out of curiosity to see if it is doable. But the point here is to remember the engineer. If you learn the biological processes leading to the production of complex molecules of penicillin and insulin, we can make them in large quantities safely for humans to use.